Hello, my name is Ian Wells. I am a senior at Winston-Salem State University in my second to last semester of clinical laboratory science program. I am presenting my research paper on the therapeutic effectiveness of COVID-19 convalescent plasma, or CCP, in hospitalized patients. This is something that I was interested in during my time as a medical laboratory technician during COVID. At the time, it was used for the late stages of a patient's care who were hospitalized because of COVID-19 symptoms. At that point, there was very little that the medical staff could do for these patients. The best thing that they could do, or what they could manage, was the symptoms of the patient until their plasma therapy, this COVID-19 convalescent plasma, came around and was used as basically a countermeasure against COVID-19 and those most affected by it. I became interested in it at that point and used it as a topic for my research paper. I was curious if at all during that time that I was issuing CCP to patients, if it was at all effective, if it helped them at all, if it had hurt them at all. I wasn't sure. I was just at that time serving in the Navy as a lab tech and just doing what I was told to do. And that is basically the basis of this research paper here that I'm presenting to you all. The abstract of this research paper is: during the COVID-19 pandemic, thousands of immunocompromised patients who were currently or previously diagnosed with lung-related illnesses such as COPD and pneumonia were met with severely diminishing health once they had contracted COVID-19. Due to the nature of the sickness, it spread quickly and left with little treatment options. The race was on for providers and researchers to develop a new treatment plan. COVID-19 convalescent plasma, or CCP, became that treatment option and hope for patients, with the intent on providing the afflicted with the necessary antibodies to defend themselves. The idea was to take patients who were not responding positively to care, the most severe cases, and infuse them with the blood product collected from individuals who had previously carried the illness, and give those antibodies to the patients who had desperately need them. So the blood product was plasma itself, taken from individuals who had already contracted COVID, and those antibodies within that product were given to patients who were in the most severe state of COVID, with the intent that those antibodies would help fight off the COVID symptoms that they were facing. That was, in my research, that was the intent of it. In that time, multiple studies across the world have conducted randomized controlled trials to prove if infusing patients with this newly developed blood product resulted in the intended result. That intended result being the positive outcome of that patient. In this case, to improve the health of severely compromised patients, based on the research that will be provided in this research paper that was pulled from multiple resources all across the internet. It can be summarized that having neither a positive or negative effect on the patients, and what proved to be a better cure was the individual's own natural ability, known as active immunity, to remedy these events. After learning that, I was a little disappointed. The CCP as a whole, just from a donor center collection perspective, it was all under lock and key. It was all very heavily monitored. And given at that time, as being active duty in the Navy, it was treated with the utmost seriousness for our leadership and within our own department. and the levels of considerations that had to be taken through our medical director, our pathologists, the patients, providers, the amount of paperwork that had to be completed to get us to the point of thawing those products and then issuing those products to patients. There was a laundry list of things that needed to be completed at first. And I'm wearing my hat backwards because one of the criteria for this assignment was to make the presentation fun, or to make the video fun, which I think is silly. So I put my hat backwards, so hopefully this counts as a full points in that category. The introduction. In 2020, during the COVID-19 pandemic, medical professionals and medical facilities alike had in an instant been overwhelmed by the volume and severity of the conditions of patients. The nature of this new condition, COVID-19, took an entire population of people who were already struggling with lung-related disorders, such as pneumonia and COPD, and put them in a more severe medical state, to the point of intubation and long-term hospitalization. Faced with this virus, researchers and scientists scrambled to find a suitable treatment option. Eventually, they turned to employ an old strategy of passive immunity. This strategy was deemed desirable due to it having been previously used during the infamous Spanish flu of 1918 by collecting blood products from patients who were exposed and possessed antibodies against the virus. It can be used in other patients who are struggling with the same virus. So, and this is a better way of explaining it that I had explained it in the abstract. Blood products of all calibers require approval from the FDA or the Food and Drug Administration, and consequently, due to the emergent behavior of the pandemic, it was given. There are a lot of controversies surrounding COVID-19. And this is outside of any political leaning or whatever. This is all research that was compiled through online, completely unbiased, and supported by the entire scientific community within North America and the European Union. The creation of COVID-19 convalescent plasma was used as a critical last-stage intervention for those individuals who were in the most critical of conditions. And that was the case at my hospital where I was working, where I was issuing it. It was only for those who absolutely most needed it. It was an exclusive exercise once all other treatment methods were offered and the condition of the patient was in the late stages of care. Unfortunately, the utilization of the convalescent plasma wasn't fully tested, as would be the case in most situations, and the full extent of known care that this blood product would or not provide was not entirely known. The purpose of this research paper is to address three of the most critical questions. Did the use of COVID-19 convalescent plasma have the intended therapeutic effects for hospitalized patients? The basis of my entire research paper. Were there any long-term concerns to those who had received it? Important part of it from my own perspective as someone who had issued the product to patients. And what was learned from this practice? The conclusion of all this, what was learned from this, from issuing this product. In this paper, I will show you that there was no therapeutic benefit or harm in those who had treated, and that those who were treated using this product were not subjected to long-term health care concerns. That was a relief to me when I learned that. The conclusion of using passive immunity as a treatment option in this case had no benefit to the patient, mainly due to it being dependent on how quickly the patient was transfused convalescent plasma before the symptoms progressed to a stage where it was too late. And I'll get more into why that is the case later in this paper. The literature review. The initial scientific literature that was available regarding COVID-19 convalescent plasma left the medical and scientific fields hopeful that there was a real solution to this devastating global issue. The most notable scientific literature published by Mayo Clinic's Expanded Access Program by Joller et al. 2020 was able to demonstrate that transfusing plasma from previously infected individual to another patient. I'm sorry, my wife is calling. I'm going to continue on. to another patient was considered safe in practice and showed no great risk in terms of side effects to that person. Due to this new information, the practice was widely adopted. However, important gaps in knowledge quickly became evident that without the randomized control groups that are typically commonplace in the research phase, could not definitively prove if the patient's innate immune response or the donor's antibodies were responsible for the recovery. The gap in knowledge proved to be a looming issue within the medical community and political landscape as well, which led to the response and more testing to answer the questions. Studies such as the recovery trial, based out of the United Kingdom, and the Conquer 1 trial, based out of the Collective of the United North America, were used to provide real data that could substantiate these claims. The aim of these trials were to compare the effectiveness of transfusing CCP to hospitalized patients and providing only standardized care to prove which pathway was most successful. So you had a group that was just using CCP as a treatment option and a group just using standardized patient care as a treatment option to determine which pathway was most successful for the patient's health. The result of these trials concluded that despite the early scientific literature that was provided, it contradicted the data that was collected. The hope of convalescent plasma gave medical and scientific professionals to find a solution to a hard-fought battle was met with a disappointing reality. The materials and methods used for this study design was based on the already existing scientific data that had been published by known publications. Using literature review information found from PubMed and Google Scholar, I was able to gather the necessary information. I used keywords in these searches such as COVID-19 convalescent plasma, CCP, randomized control trials, and hospital outcomes to find the research articles support my claim, and the claim of basically everybody else as well. The ethical considerations and rules that I imposed on myself when researching was to use data obtained from peer-reviewed trials and be large enough to provide all the information that I would need. My goal was to use fewer but stronger resources as a guide from this research paper that I knew are backed up by institutional review boards before publication could be made. I intentionally left out smaller studies and, most importantly, opinion-based articles that are often not peer-reviewed or even considered reliable in general, which I think is something that, especially during this time, during COVID-19, from the later part of 2019 into 2021, was a huge issue. The result, based on the data that was collected from the literature that was used, it provided clear results to both the safety and effectiveness of transfusion of COVID-19 convalescent plasma and hospitalized patient care. In the Mayo Clinic's program study that was mentioned earlier, 20,000 patients who had been transfused CCP had tolerated the blood product well, while less than 1% of individuals experienced some form of adverse reactions. Within my own department, we had transfused around 30 products of CCP. We've carried much more, and we issued to other medical training facilities as well, other MTFs, to support them as well. The results proved that the safety in blood product surpassed expectations and was deemed safe for continued use. After safety concerns were lifted, the next question needed answering: what was the effectiveness of convalescent plasma for therapeutic use? According to the recovery trial, that's the recovery trial was the one based out of the United Kingdom, 11,558 patients who were hospitalized were evaluated after being administered, and after a 28-day observation period, there was a mortality rate of 24%. Likewise, the individuals in the randomized control group who did not receive CP, but instead received standard hospitalized care, had a similar mortality rate of 24% as well. This concluded that per recovery, the United Kingdom trial, there were no improvement in the mortality rate from one treatment to the next. In addition to the recovery trial, the Conquer 1 trial, that is based out of all of North America, had terminated their study due early because the plasma transfusions failed to show any reduction in the death of patients, which is a similar result, but a different conclusion of the recovery trial. Discussion: Interpreting the results of the study were conducted give insight into inconsistencies between the safety of transfusing the product itself and the lack of clinical effectiveness in hospitalized patients. The explanation of why convalescent plasma failed to restore patient health is due to the mechanisms of the virus. Plasma therapy was given to individuals whose health was in the late stages and was in the end not effective because instead of the antibodies from the product reversing the effects of the sickness, it only neutralized or stopped the progression to greater extremes. During the late stages of COVID-19, systemic damages and inflammation in the lungs had already occurred, and therefore the antibodies were ineffective. The limitations found during this period of implementing a healthcare measure of this degree is based entirely on timing. If the intent of using plasma therapy was to benefit the health of the patient, then the best time to see that outcome is in the early stages of infection. Once the body enters an overacting immune response, it becomes difficult to reverse that. Another issue was that the titration of plasma or the strength of the antibody varied greatly from one donor to the other. Because of that inconsistency in the dosage of the plasma became evident. It became an issue of not knowing how many antibodies were being given to any specific patient. The conclusion of this research is during the time of the COVID-19 pandemic, the development and introduction of COVID-19 convalescent plasma, CCP, offered the world hope, but unfortunately failed to provide the intended therapeutic effects. The scientific data shows that during the period of CCP transfusions, there was virtually no safety concern for the patient. It did not cause any long-term complications to those who were transfused, but did not benefit their conditions. The primary lesson learned from this global event demonstrates to the scientific community that when dealing with passive immunity, the greatest therapeutic impact you can achieve is in the earliest stages of infection and not as a last resort. My references for this research paper are the Conquer 1 trial investigators of 2021, Escort LJ Turgan AF and McQuinton ZK of 2021, Joyner MJ Carter RE and Seinfeld JW of 2020, Ortegoza MB Yoon H and Goldfield KS of 2022, and the Recovery Collaborative Group of 2021. Thank you for your time, and I hope you learned something. Goodbye.